Hello everyone, welcome to another new stream where we are going to talk about the GATE biology exam. Let me just check if the audio and video is coming clearly. Okay, so it seems that everything is working fine. So we can start our today's discussion. So since your GATE exam is coming soon, I've decided to bring to all of you very important topics that you must revise before going for the exam. And these are compiled from the previous year questions, so they have been already asked, but the concepts or the facts that I'm going to tell you today are going to be very important for your upcoming exam as well. So that's why you should keep this in mind, and they can be mixed and matched with other facts to form new questions like match the followings or fill in the blanks kind of things. Not fill in the blanks, but yeah, numerical kind of things. So all these formulas or the facts that I'm going to discuss today are high yielding topics, and that is why you should watch it till the end and also share it with your friends if you like it. So I'll discuss some of the important facts based on the units that are important for the GATE exam, both for life sciences as well as biotechnology. So first I'll start with the biochemistry portion because it is also important for the biotechnology aspirants. So that is why we are going to discuss some of the common topics, and then we are going to focus on the biotechnology specific things at the later end of the video. So I hope the screen is clearly visible and I'm not missing some of the text here. Okay, yeah, so it's visible. So let's start with the discussion for biochemistry section. One of the most important topics is amino acids or amino acids, as some of the people pronounce it. So the first important thing that you should remember is the absorbance tryptophan, which is uh, uh, containing a ring structure. So I will show you the structure of amino acids first. Amino acids structure. The ring-like structure, the aromatic ring compounds have the absorbance in this range of 8 to 80 nanometer. This is followed by tyrosine, which is also written as Y. So this one letter code, you should also remember this. For tryptophan, it is W. For tyrosine, it is Y. And for phenylalanine, it is F. So tryptophan has the highest absorbance at 280 nanometer, followed by tyrosine. Phenylalanine also has, but very negligible in the UV range at 280 nanometer. So this thing you should remember. Let me show you the structure of amino acids parallelly so that you can revise the structures as well. So here is the structure of different amino acids. These are the basic amino acids: arginine, histidine, and lysine. Remember, the histidine has also this imidazole ring, because of which the pK becomes roughly 6. So histidine can be used as a buffer at physiological pH. So this we'll also discuss later, but just I wanted to remind you. Then these are polar uncharged amino acids like serine, it has an OH. Then threonine also has an OH. There is asparagine, which is CONH2 group amide, and glutamine also having a CONH2, but with more carbon than asparagine. Then there are hydrophobic side chains like alanine, valine, isoleucine, leucine, and methionine, which has a sulfur and CH3. So this is the ending group of methionine. Then there are these hydrophobic amino acids. So these are also aromatic amino acids. So the ones I discussed: phenylalanine, tyrosine, tryptophan. Tryptophan having the highest A280 absorbance because of this ring-like structure. But next comes tyrosine, which has this phenyl ring similar to phenylalanine, but with a OH group attached to this. So this leads to the pK value of tyrosine being around 10, which is more or less for this phenol-like structure. So this was our first fact that you should remember: the absorbance values and also the structures that I just showed you. Then structure-wise, glycine is the only achiral amino acid which has no D or L forms. Proline is an amino acid and it disrupts the alpha helix structure. So again, let's go back to the structure. So we have the glycine here. You can see there is no chiral carbon here. And proline, you can see this has an NH instead of NH2, unlike other amino acids which have an NH2 also. Here there the NH2, this ring structure fuses with the NH, leading to the formation of amino group. So this is an amino acid, remember this. And it also leads to restricted rotation around the peptide bond. That is why proline has a very restricted Ramachandran plot, which we'll discuss later. Then charge-wise, lysine and arginine are positively charged at the neutral pH at pH 7. Aspartate and glutamate are negatively charged, meaning they are acidic. And histidine has a pK of roughly 6 and acts as a buffer. So exactly what I discussed a few moments ago. So here are our basic amino acids, arginine and lysine. Histidine is roughly around 6, the pK of histidine, and then the acidic ones are uh, I guess, on the right side. Yeah, here. So aspartic acid and glutamic acid, these are the acidic ones, meaning they have a negative charge at physiological pH. Then sulfur containing cysteine forms disulfides and methionine is the start codon. So the start codon codes for methionine. Again, this table summarizes it very nicely. So you can see the cysteine having this thiol group. This can form the disulfide linkages leading to the formation of cysteine. Cysteine is the monomer, but when it forms the S kind of disulfide linkage, then it is called cysteine. And then also methionine has this SCH3. So this cannot form the disulfide because it is already linked to CH3 at the bridge. But here the SH group is free. So when this H is removed in the basic environment or alkaline environment, then this S minus nucleophile can react with other groups and can form the disulfide bridge. So we have covered this part. Then important Ramachandran plot. So I will just pull up the image for this as well. Ramachandran plot. The image is not very clear. I think this one might work. This is also slightly lower resolution. So you can see here the beta sheets have a positive psi value and negative phi value. The right-handed alpha helix has a negative psi value and a negative phi value because it is in the third quadrant. The left-handed alpha helix, which is in the opposite directions to left-handed alpha helix, which will be called like this has a positive phi value and a positive psi value because it is like a mirror image of that. So let me go back to the text and then I will describe this. Alpha helix has a negative phi and negative psi in the bottom left quadrant, which is the third quadrant if you go anti-clockwise direction. Beta sheets are in the top left quadrant in the second quadrant. Left-handed helix are in the top right quadrant, which is example of this is the glycine. Glycine can be present in all the four quadrants. So you can see the left-handed alpha helix is allowed only for some flexible amino acids like glycine, whereas it is disallowed for most of the other amino acids. Proline has a very restricted Ramachandran plot region, and remember this collagen triple helix is also here. Parallel sheets are here, and anti-parallel sheets are slightly above them. So they have a slightly higher psi value than the parallel beta sheets. So this is something that you should remember just as a fact because it can be asked to identify which of these regions correspond to which type of protein secondary structure. So then you should be able to identify. There is more information about different types of helix helices and different types of allowed or disallowed regions. So remember these are the allowed regions. And there are some partially allowed region which is not shown in this figure. Perhaps this one is more clear. So what you should remember here is this region where are the beta sheets found, where are the right-handed alpha helix, and where are the left-handed alpha helix found. I will also show you this plot for glycine. For glycine, and you will be able to appreciate that for glycine the region is much more extended. So glycine can be found in different regions. See, for glycine it is present here as well in the left-handed alpha helix form. Yeah, so this is for glycine and this is for proline. So proline has a very restricted region as compared to other amino acids, whereas glycine has a much more flexible region, so much more allowed region as compared to the standard amino acids. Although glycine is itself a standard amino acid. Then collagen, remember, contains modified amino acid like hydroxyproline, so proline with a hydroxy group, hydroxylated, and it has a triple helical structure where this repeat is very important, glycine XY. So this can be hydroxyproline and proline. This kind of amino acids are present there. Then coming to enzymes, remember the zymogens are inactive precursors. For example, trypsinogen getting activated to form trypsin via enterokinase. So this reaction you should remember what are zymogens. So the inactive form of the enzyme is called zymogen. And isozyme are a different structure having same function. For example, LDH. LDH can have different structure, or enzymes like LDH can have different structure, but they can form the same function. Again, very important one is coenzymes or vitamins. So, for example, here B1 is thiamine and it is involved in decarboxylation reaction. B2 is riboflavin. It is again involved in redox reaction present in FAD and FMN. Flavin adenine dinucleotide. If you remember the name, you can see there is riboflavin, and this is also flavin adenine dinucleotide. Here it is thiamine pyrophosphate TPP. So thiamine, if you remember the full form of these coenzymes, then it will be very easy for you to identify which uh, vitamins are present in that. Then there is niacin. So this is nicotinamide adenine dinucleotide, and this is nicotinamide adenine dinucleotide phosphate. Again, very standard in respiration. You might have read about NADH and NADPH in photosynthesis. So niacin is required here, which is vitamin B3. Remember this also, which is B1, which is B2, and which is B3, and all this. B1 is thiamine, B2 is riboflavin, B3 is niacin. Then B6, again very important, pyridoxine. So this is involved in transamination reaction, and B7, which is tetrahydrofolate or biotin, it is involved in carboxylation. So I will show you a table for this as well. Meanwhile, let me check if there are any questions. Yeah, so here you can see niacin is nicotinamide adenine dinucleotide oxidation or hydrogen transfer, riboflavin, flavin adenine dinucleotide. This is not a sequential order. I think this one is better. Let me make it bigger, this image. Slightly blurry, but I think it's visible. So B1 is thiamine pyrophosphate decarboxylation. B2 is FMN, like B2 is involved in the structure of FMN, riboflavin. B3 is niacin and ADP and ADPH. B6 is pyridoxyl phosphate, amino group transfer or transamination, as I told you. B12, very important, cyanocobalamin. So cobalt is very important here. So the coenzyme is methyl cobalamin or deoxyadenoxyl cobalamin. Just remember cyanocobalamin, vitamin B12 for rearrangement reaction, biotin, carboxylation reaction, folic acid, tetrahydrofolate, carrier of one carbon unit, and pantothenic acid, coenzyme A. So for coenzyme acetyl coenzyme A, you require the pantothenic acid, which is carrier of acyl group. All these things, then name and the function you should remember. Here again, it is nicely summarized. So I will share it with all of you later on. But for now, you can focus on the key structures and the mechanism that they have. I'll share it with you later on. Don't worry about that. Then comes metabolism and inhibitors. So here we have glycolysis. The rate limiting enzyme here is phosphofructokinase 1, PFK1. TCA cycle occurs in the mitochondrial matrix. Always remember this. It happens in mitochondrial matrix. TCA can't happen in the cytoplasm. Glycolysis happens in the cytoplasm. Here the rate limiting step is isocitrate dehydrogenase. So remember this as well. This can be directly asked, which is the rate limiting step of TCA. It is isocitrate dehydrogenase. Then some of the inhibitors, very important, remember them. Complex 1 can be inhibited by rotenone, complex 3 by antimycin A, complex 4 by cyanide, carbon monoxide and azide, which is cytochrome C oxidase. So this can be blocked. So and it has been asked repeatedly. ATP synthase can be blocked by oligomycin. Don't get confused between complex 3 and ATP synthase because the names are similar. They have mycin, but the anti-mycin one can block complex 3, but ATP synthase can be blocked by oligomycin. So remember, like the ATP synthase has this channel. So remember it like oligomycin. O is like that opening of that. So it will be easier for you to remember oligomycin, ATP synthase, which has a channel F0, F1 particle. Uncouplers example is dinitroxenol, thermogenin. This allow the proton flow without the ATP synthesis. So the ATC will still take place, but there will be no formation of ATP because the proton gradient is broken. Now coming to microbiology, remember 16S rRNA. This is very important. 16S rRNA is used for phylogenetic classification, and this system was given by Carl Lewis. Then for cell wall, gram-positive bacteria have thick peptidoglycan wall, thick peptidoglycan. That is why they take up the gram stain plus teichoic acid. And gram-negative bacteria have thin peptidoglycan. That is why they do not take up the gram stain. Plus they have LPS, lipopolysaccharide, which acts as endotoxin, and they have an outer membrane as well. So remember, that's why they are much more deadlier and more pathogenic, the gram-negative ones. Then for archaea, remember the walls of archaea are ether-linked lipids. There the structure is the ester, but here the structure is ether-linked and monolayer in some, and there is no peptidoglycan. That is why it is called pseudomurine. Pseudomurine, remember this in archaea. Then some of the antibiotics and their mode of action. For example, cell wall synthesis inhibitors, for example, the family of penicillin, ampicillin, vancomycin, all of these block the synthesis of cell wall. Remember, not cell membrane, cell wall. So this peptidoglycan wall, which is made up of these units of N-acetylglucosamine and muramic acid, this is not allowed to be synthesized because of this beta-lactamase enzymes. Then protein synthesis in this smaller subunit, 30th subunit, can be inhibited by streptomycin and tetracycline. This you should remember directly. There is only one way, which is to revise it multiple times. In the larger subunit, chloramphenicol and erythromycin, RNA polymerase can be blocked by rifampicin. Remember this, very important. RNA polymerase can be inhibited by rifampicin in the bacteria, and then DNA gyrase can be blocked by quinolones like ciprofloxacin. Then nitrogen fixation, the enzyme is very important here. The nitrogenase enzyme, which is a molybdenum, it has molybdenum and iron. Remember these two ions. Protein highly sensitive to oxygen, and that is why it requires leg hemoglobin to protect the enzyme and allow it to function properly. It requires leg hemoglobin, which scavenges the oxygen. Then the reaction is N2 to 2NH3 requires 16 ATP per nitrogen. For one nitrogen to be fixed to 2 ammonia, you require 16 ATP. You can also look at the balance reaction here. I think around 8 protons are also required. But the important thing is how much ATP is required. This you should always remember. Then for sterilization technique, autoclave is done at a temperature of 121 degrees Celsius at this much temperature, pressure, and for 15 to 20 minutes. This is called moist heat because you are having a higher pressure steam and then it is also heated. This kills the spores. So spores are the resistant form of the bacteria which can be transmitted for multiple generation when the conditions are unfavorable. So how do we ensure that even those forms are killed? That is why we require autoclave at a very high temperature. And pasteurization is done at 72 degrees Celsius, which is lower than autoclave for 15 seconds and does not kill the spores. This is important. Pasteurization does not kill spores. Then filter membrane, if you have a 0.22 micron pore size, then you can filter the bacteria, and if you have 0.02 micron, then you can filter the viruses. Now coming to the third section, which is biotechnology and methods. Here the recombinant DNA technology is very important. So remember the restriction enzymes which are type 2 are generally used, and they cut within specific sites which are called the palindromic sites, and they require the magnesium ion. So like most of the molecular biology enzymes, they also require magnesium ions. Then here is an important question that can be directly asked: the vector cloning capacity. So yeast artificial chromosome has the largest capacity, 1000 kb, followed by bacterial artificial chromosome. Then comes the cosmate, which is roughly 45 kb. Then comes phage, and smallest among all of them is plasmid. Remember this sequence: plasmid, phage, cosmid, back, yak. This is very important. Then PCR steps, you should always remember: denaturation, annealing, and extension. DAE, DAE, always remember. Denaturation is done at a very high temperature, around 95 degree. It can be 92, 93 also. Annealing at 50 to 60 degree, slightly lower temperature, and then extension at 72 degrees Celsius using Taq polymerase. You can also use PFU, which is Pyrococcus furiosus polymerase, but generally in the textbook Taq polymerase is mentioned, which is Thermus aquaticus polymerase resistant, which is a thermostable polymerase. Then coming to immunology, so antibodies, you should remember IgG is the most abundant and it can cross placenta. It is also seen in the secondary response. IgM, it's a pentamer and it is first produced. So for primary response, IgM is involved. For secondary response, IgG is involved and it is also produced in the highest quantity. IgA is a dimer and it is found in secretion like tears, milk, like colostrum, which is produced by the mother, and gut. And Ig is involved in allergy and parasitic infection. So E allergy, you can remember this. MHC classes, MHC class 1 is present on all nucleated cells, and this leads to the recognition of CD8 positive T cell, which are cytotoxic. Then MSC class 2 is present only on the antigen presenting cells. Remember this, only on antigen presenting cells. Example, dendritic cells, B cells, macrophages, and they help in the activation of CD4 positive T helper cells. Then hybridoma technology, fusion of B cell and myeloma cell. B cell and myeloma cell. B cell which produce the antibody, and myeloma cell which has higher division capacity. So when you fuse both of them, it is called hybridoma, and using polyethylene glycol. So this is used as a fusing agent, fusogenic agent. Selection media used here is HAT, which is hypoxanthine aminopterin thymidine. So this is again very important. You should read about this, why it is used. I'm just giving you a brief summary of all the facts that you should quickly remember. Then coming to molecular biology, there is DNA replication enzymes, very important. DNA Pol1, it removes the RNA primers. Once the RNA primers are laid down, you also have to remove them and seal the space. You don't require a RNA DNA hybrid, right? So you have to remove that. So how do you do that? You use DNA polymerase 1, which has the 5' to 3' exonuclease activity and fills the gap. So it removes the RNA primer and then it also fills it in the 5' to 3' direction. The Klenov fragment, this is very important, remember this. The Klenov fragment lacks this 5' to 3' exonuclease activity. If someone asks you, is the exonuclease activity present in the Klenov? It's not present, just remember this. So someone is saying thank you, no worries, you can share it with your friends as well and they can also join. There is one more question, will you provide the zoology revision portion also? Today I'm mostly going to cover molecular biology and other topics, microbiology, biochemistry. I have not prepared the notes for zoology, and although I think I have already covered most of it in one of my lectures, so I can link it in the comment section later on. If you want to revise, you can revise it from there. So most of the questions you can cover from there. But I'm not going to cover zoology separately in this session. Then there is another enzyme, primase. So it is an RNA polymerase which synthesizes RNA primer. So remember, primase making RNA primer, and it does not require a 3' OH to start. So this is very important. Had it been the other case that it requires a 3' OH, then there would be no primer synthesis at all. Because how will we have a 3' OH to start with, right? On the other strand where there is nothing, the RNA primase has to start without the 3' OH in a random position, and then from there on you will have the 3' OH which the DNA polymerase can extend. Using that DNA polymerase, the 3' OH, the DNA polymerase can extend and form the DNA. That is why it is very important that primase can form the RNA primers without the 3' OH. Then there is telomerase, which is a reverse transcriptase. You should also remember what is reverse transcriptase. It is RNA dependent DNA polymerase. For example, in viruses, in HIV virus, you have the RNA genome. So how do you convert it to DNA in order to integrate with the host? You do the reverse transcription using the enzymes like reverse transcriptase. So telomerase in our genome is also a kind of reverse transcriptase. So using the RNA template, it makes the DNA. It carries its own RNA template. You should also remember the sequence for this. Then there is topoisomerase. So type 1 topoisomerase cuts only one strand of DNA and it does not require ATP. The type 2, which is also called as dirase, cuts both the strand and requires ATP. So this distinction you should also keep in mind. Then there are transcription factors in prokaryote. For example, there is sigma factor, which recognizes the promoter. So there are regions in the promoter, for example, -10 region, which is startup box, and then there is the -35 region. So minus upstream of the promoter of the transcription start site, there is -10, which is startup box, and -35, which is another region. So this is recognized by the sigma factor. And sigma 70 is the housekeeping factor, and sigma 32 is for heat shock. So whenever the bacteria wants to express proteins which are important in heat shock response, then it will recruit or it will make more of sigma 32, and that will lead to the expression of genes which are involved in heat shock. Because there will be more amount of sigma 32, so that can bind to the RNA polymerase, and specifically it can identify the regions which are going to express the heat shock proteins. Sigma 70 is the housekeeping factor, so that is present in general without any like specific function. Then there is rho factor, which is required for rho dependent termination. So there are two ways of termination: rho independent and rho dependent. For rho dependent, you require this rho protein as well, which binds to the rut site on the RNA. So the RNA also has this rut site where this rho factor binds, and then quickly pulls out the RNA from the RNA polymerase complex, leading to the termination of the transcription. And coming to translation inhibitor, so here the match the following kind of questions are very common. So remember this, similar to what I had told you about the different antibiotics. Here you should also remember the translation inhibitor. So for example, puromycin. So puromycin mimics the amino acid tRNA synthase, and this causes the premature chain termination both in eukaryotes and prokaryotes. Remember also the host. So puromycin can be used both for prokaryotes and eukaryotes because it mimics the tRNA, amino acylated tRNA. Next is cycloheximide, and it inhibits the eukaryotic ATH ribosome. This is again important. So it inhibits the translocation cycloheximide. Then there is diphtheria toxin, which inactivates the eukaryotic elongation factor 2. Eukaryotic elongation factor 2. That is why diphtheria is again toxic for eukaryotic system via the ATP ribosylation. And then there is alpha amanitin. This inhibits the RNA polymerase 2. So the one that leads to the expression of mRNA, that is blocked, found in Amanita phalloides, which is a death cap mushroom. So if someone eats this mushroom, they will also ingest this alpha amanitin compound, which will lead to the global shutdown of mRNA and thus leading to the death of the individual. Then coming to the bacterial operon, so one of the most common one is lac operon, which is having a negative control by the lac inhibitor repressor, lac I repressor, and then there is positive control by the cap cyclic AMP site. And then when glucose is present, the cyclic AMP is low. There is no cap binding, and there is also low transcription. But when lactose is present, then allolactose can bind to lac I site. This can have the repressor shut down, so the repressor goes, and then the transcription can start for the structural genes, which are Z, Y, A. So Z is for beta galactosidase, Y is for transacetylase, and A is for Y is for permease, and A is for transacetylase. So this you should remember. Then comes tripoperon or tryptophan operon. It has the attenuation mechanism involving a leader peptide. So the leader peptide is rich in tryptophan, and higher tryptophan leads to terminator hairpin forms. So the terminator hairpin structure, 3 or 4 loop, can lead to the stopping of the transcription. Whereas when there is low tryptophan, then it can lead to the formation of entire expression of entire tripoperon genes. You should also remember the sequence. So A, B, C, D, E. This is in reverse sequence. Let me just show you the picture of this tryptophan operon. See, so this is the structure. Maybe I will open another one. Yeah, so you can see the structure here, the genes of operon A, B, C, D, E. These are the proteins that are synthesized from this. And this is the TRIP-R region, and the promoter is upstream here, and the promoter for the structural genes is somewhere here. So this is how you should remember that tryptophan is absent, the repressor is inactive, and the operon is on. So if there is more tryptophan, then the repressor will become active, and then the operon will be shut down. So the mechanism here is attenuation mechanism. Remember this. Next, coming to cell biology and signaling. So here the cytoskeleton drugs, again some of the inhibitors and drugs you should remember. So colchicine and vinblastine inhibit the microtubule polymerization, and this can be also used to treat cancer, tumor. Then Taxol, again for treating tumor, Taxol or paclitaxel inhibits microtubule depolymerization, so it stabilizes them and preventing cell division. So here it stops the cell division, Taxol, by preventing depolymerization, and colchicine stops the polymerization. Then cytocalycine disrupts the actin filament. So both of them work on microtubule, but cytocalycine works on actin, which is microfilament. Remember this. Cell cycle regulators. So MPF, maturation promoting factor, which is cyclin B plus CDK1. This we should remember, very important. What is MPF? Which drives the G2 to M phase transition. Then P53, as all of us know, guardian of the genome, triggers apoptosis if DNA is damaged and activates P21. So P53 activates P21. P21 leads to the apoptosis. Then RB protein is also tumor suppressor protein, so it binds to E2F transcription factor. When RB is phosphorylated by CDK, then E2F is released and S phase genes are expressed. So to stop the cell cycle, RB should bind to E2F, but if E2F is phosphorylated by the CDKs of the S phase, then RB will be dissociated from the E2F, and E2F can go to the nucleus and lead to the expression of S phase genes, which will lead to the transition. Then there is signaling pathways. So GPCR, this is very compressed way, but you should at least remember these many facts for the GATE exam. For NET exam, I will go in a much higher depth, but for currently, you have very less time also. So I'm just telling you most important things here for your exam. So GPCR is 7 transmembrane helical structure. It has alpha subunit, which hydrolyzes GTP to GDP to switch off. If GTP is hydrolyzed to GDP, the switch is turned off. So in the presence of toxin, what happens? The ADP ribosylates GS alpha. So the G stimulatory alpha is ribosylated, ADP ribosylated, which leads to permanently active adenylate cyclase. When the adenylate cyclase is permanently active, there is high cyclic AMP. High cyclic AMP leads to the loss of water. In the case of pertussis toxin, what happens? Again, ADP ribosylation, but of G inhibitory, GI alpha. So it cannot inhibit adenylate cyclase, meaning again there is high cyclic AMP. So here the cyclic AMP is high because the GS alpha is always active. And here the cyclic AMP is high because the G inhibitory alpha is inactive. So this is how both of the toxins work, the mechanism. Then there are analytical techniques for separation. Briefly, let's go through them as well. Size exclusion chromatography, also called as gel filtration chromatography. Here, the large molecules comes out first, elute first, and cannot enter the bead pores. Small molecules have to go a longer path, have to travel a longer path, and they elute last. Then there is ion exchange. So remember cation exchange, like carboxymethyl cellulose. Beads are negative. It is cation. You are exchanging the cation, so the beads have to be oppositely charged. So the beads should be negative, and it binds to the positive protein. So the protein here is cationic because of the pH that we adjust. And how can we elute it? We can elute it with high pH or sodium chloride with salt. So the electrostatic interaction between the beads and the protein will break if we increase the salt concentration. In an ion exchange, it is some ethyl ammonium salt, DEAE. Beads are positive because you are exchanging anion, so you need the beads to be positive so that it can attract the anionic protein. It binds to the negative protein. Then in affinity chromatography, you use specific ligand, example GST tag for glutathione and His tag for nickel NTA. So this again you should remember. Then coming to electrophoresis, there is SDS page which separates by molecular weight only. The SDS in the SDS page masks the charge, which means all the proteins will be first denatured, so they will be linearized and then they will be masked with the negative charge on them. The reducing agent beta mercaptoethanol or DTT can break the disulfide bond to linearize the protein or denature the protein. Then there is isoelectric focusing, which separates by charge. Remember this. So SDS space separates by molecular weight or mass, and isoelectric focusing separates by charge or the pi of the protein. Protein stops moving when pH equals to pi. So when pH equals to pi, the protein charge becomes neutral and the protein will not move because there will be no effect of the electric field. Then coming to some specific immunological techniques. So here remember the hypersensitivity types. First is type 1 hypersensitivity, which is mediated by IgE. Remember this. This can lead to anaphylaxis or allergy and also mast cells degranulation. Then there is type 2 hypersensitivity, which is done by antibody mediated cytotoxicity. And this happens usually in the blood transfusion reaction when there is RH incompatibility. Then there is type 3 hypersensitivity, which is immune complex mediated, and it is examples are serum sickness and SLE or lupus. Then type 4 is delayed or cell mediated. Again, here the example is TB, skin test, and contact dermatitis. Coming to vaccine adjuvants, so alum, which are basically aluminum salts, they are most common human adjuvant and promote the T helper to Th2 response. And then there is FC or Freund's complete adjuvant, which is containing the heat kill mycobacterium, and it is used in animals only. So this was about the important facts. Now we'll also look at some of the important formulas. But before that, let me have a look at the chat section and see if there are any questions. Okay, so let's move ahead with some of the important formulas that will be directly asked in your exam in the form of numericals or in the form of match the following or simple match single correct options. So that's why you should remember this formula. First one, enzyme kinetics, very, very important Michaelis-Menten equation. V naught equals to V max times substrate concentration divided by Km plus substrate concentration. Remember, the substrate concentration, when it equals Km, then the V naught will become equal to V max by 2, half of the maximal velocity. So when the V max by 2 velocity is achieved, the Km equals to that substrate concentration. Then the turnover number kcat is defined as kcat equals to V max over Et, which is the total enzyme concentration. Another thing can be asked is efficiency. So this I also solved in one of the previous year questions while discussing. And the efficiency can be written as kcat, that you calculate here V max by Et, and you divide that by the Km, then you get the efficiency of the enzyme. Then some inhibition patterns in the Lineweaver-Burke plot. So here we have for competitive enzymes, Vmax will remain same, Km will increase. For non-competitive, the Vmax will decrease. So the maximum velocity that the enzyme can achieve will decrease, but the Km will remain same. So there is no effect on the affinity because the inhibitor is not competing with the substrate. And then there is uncompetitive, both will decrease. So the Km will also decrease, meaning the affinity will improve, but the maximal velocity that can be achieved by the enzyme will decrease. We will get parallel lines in the Lineweaver-Burke plot. Then microbial growth kinetics, here you can see ln nt over n0 equals to mu t, or it can be also written as this, nt equals to n0 times e raised to mu t or kt, whatever you remember. Mu is the specific growth rate. Then doubling time can be simply written as ln2 by k or ln2 by mu. ln2 is roughly 0.693 or 0.7 if it is easy for you to remember. Then the Monod equation. So Monod equation is exactly same as the Michaelis-Menten equation here. Just replace the V max and V with the specific growth rate. So mu equals to mu max times concentration of the bacteria divided by Ks plus concentration of bacteria. Exactly same as the form is exactly same, but the interpretation is slightly different. So here it is called the Monod equation. This is the concentration of substrate, not bacteria. Then there is bioenergetics and thermodynamics. So we have delta G equals to delta H minus T delta S. And delta G can be also written as the sum of delta G naught, which is at standard condition, in this standard condition, plus RT ln K equilibrium or ln Q. Then we have at equilibrium, the delta G becomes 0, meaning delta G naught, this can be transferred on the left side, is equals to minus RT ln K equilibrium. So here R equals to this and temperature must be in Kelvin. Remember this while solving the numerical. Then there is Nernst equation. This is again very important, although most of the questions can be solved up to this point. But if in case the examiner asks you about electrochemistry as well, you should remember this delta E equals delta E naught, similar to this form, minus RT over NF ln K or ln Q, whatever it's easier for you to remember. Just remember this, minus RT over NF. F is the Faraday constant, N is the number of moles of electron, R and T are temperature and universal gas constant, same as above. Then there is ln Q or ln K, equilibrium constant. Coming to genetics numerical, so we have for 2 alleles, P plus Q equals to 1. If there are more alleles, P plus Q plus R plus S, whatever equals, sum should be 1, frequency of all the alleles. Then P square plus 2PQ plus Q square is also 1. So if you just square this, left hand side RHS and LHS become 1. So here the P square is the frequency of homozygous dominant, Q square for homozygous recessive, and 2PQ is for heterozygotes. Finally, coming to spectroscopy, Beer-Lambert law, very important. Absorbance equals to epsilon LC or molar extinction coefficient times concentration times path length. This you should remember. Very easy numericals can be asked based on this. And then there is yield coefficient, which is basically mass of cells produced divided by mass of substrate consumed. So how many cells produced, the mass of those divided by how much of the substrate is consumed. Minus, because it is reducing, dx by ds. So substrate concentration has reduced, so that's why you take the minus of that to compensate for that. Then there are some advanced formulas for the bioprocess engineering. So The first two parts are important for life science students and other subjects. But for bioprocess engineering, people who will be targeting this biotechnology, here are some of the important formulas that you must know before going for the exam. There is the Del factor for sterilization kinetics. You should know this, which is a measure of sterilization effectiveness, how effective your sterilization is, which is again similar to the growth equation here, nt equals to n0 e to the power mu t. So you can just remember it like this. N0 is the initial spore, nt is the final spore, which is usually very low, like 1 in 1000 chance, is the specific death rate. So instead of growth, you are talking about death. Then bioreactor scaling and oxygen transfer. Here the oxygen transfer rate is written as KL times A, which is volumetric mass transfer coefficient, followed by the product of saturation solubility of oxygen minus actual dissolved oxygen concentration. Saturation minus actual times the volumetric mass transfer coefficient. So this will give the oxygen transfer rate. Then this is again very important, Reynolds number for stirred tank reactors. Reynolds number can be written as n d square rho by mu. So the n is the impeller speed, then d is the impeller diameter, rho is the density divided by the viscosity. There is other form also, V, I remember it like this, rho V d square by eta. So some people write viscosity in terms of eta. Here it is given in the terms of mu. So depending on the textbook it can change, but you should know what is what, which values are in the numerator and which values in the denominator. You can, I remember it like this. V rho Vd square rho V d square divided by eta. You can also remember it like this if you find it easier. Then for centrifugation, remember the Stokes law, velocity of settling velocity is equals to d square times the difference in the density times g divided by 18 eta. You can write here eta. So this is rho g and d square divided by 18 mu. This is the settling velocity. Velocity increases with the square of the particle diameter. If the particle is becoming larger, the velocity will be also higher. Then filtration rate of filtration is directly proportional to the pressure drop and inversely proportional to viscosity and resistance. So you can also write this in the form of equation. Rate of filtration equals to delta P over eta times resistance. And then thermodynamics, here we have the Fick's first law, which is J flux, J equals to minus D diffusion coefficient multiplied by del C by del X, change in concentration with distance. It is the diffusion coefficient, and minus n indicates the movement from high to low concentration. So this was all some of the important things that I could find from the previous year questions. I would also share this in my Telegram group, and if you're new to this channel, do subscribe this and also share it with your friends. And I will also try to share some of the important figures in the Telegram group. So if you're new, follow and subscribe this channel and also share it with your friends. I will see you in a later one, and all the best for your exams. Do well, keep calm, and most of the things will be from the things that I covered here. So if you just revise it properly, you will be able to attempt the exam very nicely. So with that message, take rest and all the best.